Yeah, so hello everybody. Welcome to our optical science colloquium. It's very nice to see so many of you here today in the room and also so many of you on Zoom. So today we have a very great speaker from Germany, Professor Vahid Sandohgar. Professor Sandohgar is a director at the Max Planck Institute for the Science of Light in Erlangen. And he's also an Alexander von Humboldt professor at the University Erlangen-Nuremberg. Vahid is actually one of the pioneers of nano-optics, and this is a field that investigates light-matter interactions at the nanometer scale. And he is the founder of the Max Planck Zentrum für Physik und Medizin. And this is basically like a joint research center that combines fundamental medical research with physical and mathematical methods. Vahid obtained his PhD in atomic physics from Yale. And after that, he was a postdoc at the École Normale Supérieure in Paris and at the University of Constance. And before he moved to Erlangen on this very prestigious Humboldt professorship, he had a professorship at the ETH Zurich. Vahid won, of course, like many prizes. Too many prizes to tell you all here, but one prize that actually sticks out is the prize for quantum electronics and optics from the European Physical Society. Vahid, we are very happy to have you here today, and the stage is yours.
No,
okay,
so
no. So Vahid,
now you hear me?
The stage is yours.
Thank you. Thanks very much, Florian. It's a great pleasure to be here. I usually don't cross the pond just for a lecture, but I was very eager to come to Arizona, to Tucson, because this is one of the hotspots of optical sciences, and I didn't want this opportunity to let go of this opportunity. So I've had several discussions and meetings, and I've learned quite a bit this morning, and I'm looking forward to learning more tomorrow. So I've tried to put a colloquium together, and I was told that it should, as usual in a colloquium, I have to give more of a general introduction. But as you will see, our work is, from a fundamental point of view, extremely simple. So anyone who's here past first semester physics should be able to follow everything that I tell you. And if you have questions, then you feel free to interrupt me in the middle. I'll be happy to walk you through whatever you didn't follow. But before I start the science, since I come from far away and Erlangen is not necessarily a well-known place in Germany. And in fact, I was just asked whether Erlangen was part of former East Germany or former West Germany. So I thought that I would show you a little bit where Erlangen is. This is Germany. Places that you would know would be Berlin, Frankfurt, Munich, Hamburg. Erlangen is right here. It's about 2-hour distance to Munich or to Frankfurt. It's a town of about 100,000 people, but seat to many global companies, including Siemens and Siemens Healthcare, and you might be wearing products from Adidas or Puma. They're all basically in Erlangen. But the most important thing in Erlangen is our institute, of course. This was established in 2009, so it's still fairly new, one of the newest Max Planck institutes. And the building is from 2016. If you happen to be in Germany, please do come and visit us. And Florian mentioned briefly, we have now initiated a new effort. This is another building that is being inaugurated this year on the campus of the University Hospital. And it's an annex of our building in a way, or of our institute, and the goal is to put a whole bunch of physicists and mathematicians into this building right next to medical doctors and try to understand what their problems are so that we can try to address them. And my own group, its kind of scientific past is anchored in more quantum optics and near-field microscopy and near-field optics, but we have moved to be very, very interested in problems of biophysics and medicine, and you will see a little bit of that in my talk. But let me start with something very simple, and this is as complicated as the talk will get. But it's a problem that is actually very simple and very subtle at the same time. And I encourage you to kind of go think about it more or read about it more if you get a chance. So the problem is as follows. You have an object and you illuminate it, and then you detect along the same path. So you're illuminating along with a well-defined K vector, and then you put your detector right along that line of sight. And you know that you're going to detect less light because some of the light is going to be scattered, for example, by this object. And you also know that some of it might be absorbed by the object. But from an energy point of view, you know that you're going to have less light in the forward direction. And the power that would be associated with this interaction could be written as the incident power times some kind of a sigma, which is kind of a phenomenological area, divided by the area over which you had confined light. This is a very, very basic kind of thing that you've learned in your lectures. Now, it turns out that when you look into it and do this more rigorously, this is actually an interference problem. And they're both completely equivalent. So you can look at this as an energy problem, or you can also look at it as an interference problem. In the interference problem, you're coming with plane waves and you're scattering like a dipole, let's say if this is a small particle. And these two fields are coherently adding up in the forward direction and interfering with each other. And the phase turns out to be such that you get destructive interference in the forward direction. And that's kind of the same thing as saying you have less light if you were to put your detector right here. This is a kind of a very simple way of looking at it, because when it comes to phases and all of these things, the problem can become quite subtle. And this is typically introduced under the title of optical theorem in any kind of a problem context that involves a scattering, whether it's quantum mechanics or light or X-rays or whatever. So the intensity that you detect at your detector in the forward direction is going to be the sum of the two fields squared, which means that if you expand it, you're going to get something like this. So you have the incident intensity, you have the scattered intensity, and then you have the cross term, and this cross term is called the extinction term. All right, so now let's say that your object is very small, so that the scattering is Rayleigh scattering. And again, we know from our elementary electromagnetic theory that the electric field that's scattered is proportional to the electric field that is incident, with a proportionality factor that is called polarizability. And this is the expression that you would have for the polarizability of a spherical particle. There are a few parameters that are involved. There's obviously the index of the medium and index of the particle. But more importantly, what you probably don't care about when you go through this is that it's proportional to volume. So polarizability has a unit of volume. And this is actually quite important when you think about sensitivity limits and things like that, that I will be dwelling on for the next few minutes. Now, polarizability is also the fundamental property that goes into the scattering cross-section and absorption cross-section. Of course, because the dielectric function or the index of refraction are complex quantities, polarizability is also a complex quantity, so it has a real and imaginary part. And the imaginary part is what gives you the absorption, and the absolute value is what gives you the scattering cross-section. So you can kind of go back and forth between these concepts in different contexts. Now, the point that I want to kind of emphasize is that even in a normal kind of imaging conditions where you look at a shadow or where you do bright field microscopy or imaging or anything, what you have is an interference phenomenon, because what you're doing is if you have an object, you illuminate it, and if you were to write the electric field at this point, you could write it as the electric field of the incident light plus the electric field that interacted with this object and got to that point. And again, you would have this kind of equation. Now, it becomes interesting for us who are interested in nano-optics to ask the question, what happens if I shrink this teapot to a single teapot molecule? And the answer is, well, I'm no longer going to see the shape of the molecule or the shape of the teapot because of the diffraction limit that I have at optical frequencies, but I will scatter some light from it, so I will see a shadow from the single molecule. And because there is some light that is being taken out or scattered by this single molecule. Now, if you have a single molecule, or let's say if you have a single atom that can be approximated as a two-level atom, the way you do in your quantum mechanics course, then the cross-section, the extinction cross-section of that atom can be derived in one page, simple calculation, to be essentially lambda squared divided by 2. So lambda is the wavelength of transition from the ground state to the excited state, and it's typically in the range of a few hundred nanometers, right, in the visible range. So lambda squared is actually a pretty large area that is the cross-section of a single atom or a single molecule, okay? So when you send your light onto a single molecule, you could say that it's just a single molecule, so I'm not going to see the shadow of it because it's too little for it to make a difference. On the other hand, if you think about its cross-section, you say, well, but this cross-section is not that little. I can focus my light down to something of the order of lambda. So I should have something that is of the order of 1 if I think about sigma over A, the formula that I showed you in the beginning. And indeed, we've shown in an experiment that is now more than 10 years ago that you can detect a shadow of a single molecule. What you see here is a shadow of a single molecule. So the experiment was done as depicted here. You have molecules that are sitting kind of apart from each other on a surface, and we focus light through this sample and collect the light with another objective and send it to a detector. I'm simplifying the detection process. You have to be somewhat careful to make this measurement because the shadow that I'm seeing is of the order of part per million of the intensity that I'm launching onto the molecule. So I send something like a microwatt and I detect a picowatt of loss when a single molecule comes in my beam, okay? Now, it's detectable, but of course it's a small effect. So the thing to kind of notice or to remember or be impressed by is that the signal that you get is coming because some light was scattered by the molecule. So you might think of this as an absorption process, but it's the light that is scattered by the molecule that interferes with the light that was incident to give you this dip, if you think in terms of that interference term. Now, that was for a single molecule, but if you now back out and say, okay, I have a nanoparticle, this is going to become the topic of the next 10 slides or so, which connects to our more biophysical type of research. This technique is called interferometric scattering microscopy, in short, ISCAT. And the first time that we reported this was in 2004. And we were working on this because we were trying to detect very small gold nanoparticles. And at the time, in the late 90s, early 2000s, there was a lot of interest in detecting very small gold nanoparticles for different reasons. This was basically the beginning of what you would today know as a field of plasmonics. And a gold nanoparticle is kind of like the smallest plasmonic unit that you would have. And we wanted to detect, do plasmonics at this fundamental level. So you wanted to detect very small nanoparticles and do spectroscopy on it and all that kind of stuff. But there was also a general interest in detecting very small particles without using fluorescence, because fluorescence microscopy at the single molecule level was reported in the beginning of the 90s. Many of you were not born yet, but it's really not that long ago. That's when I got my PhD. So that's not that long ago. And during that time, that is since the time that I got my PhD, that was when people began to see single molecules, but via fluorescence. But there are only a few species in the world that actually fluoresce. So it's not the most universal way of detecting objects. So you wanted to detect things possibly via its scattering, because everything scatters light. Now it scatters maybe only a little, but everything scatters, but not everything fluoresces. So this was kind of the background of this type of work. And so in this work, we reported detection of very small gold nanoparticles down to 5 nanometers. But more importantly, we quickly realized that we can detect any other kind of nanoparticle as well. And in particular, we could detect things like viruses. Because a virus, now after the pandemic, everyone knows viruses and knows that they are kind of nanoparticles. A coronavirus is about 100 nanometers or so, you would know. The virus that we were working on was about half the size. And we were doing a very simple experiment. We were putting the virus at a cover slide. And now what you're doing is you're illuminating this. And instead of detecting in transmission, we're detecting in reflection. So you're detecting the light that is scattered by the virus in the reflection arrangement. And because there is an interface here, there's a little bit of light that is also reflected here. And these two electric fields interfere, just as they did before in the forward direction, now in the backward direction. But it does the same thing, and it gives you a scattering signal. And this is now one of the first videos that we were recording of a single virus moving as a kind of a Brownian motion on a lipid membrane. So this is the cartoon. The virus is docked to a lipid membrane via its receptors. But because the lipid membrane is a fluid kind of carpet, the virus moves around, and then we're detecting how it's moving. Yes, you have a question?
Is there a Zoom issue that you have a
No,
from the
I
floor?
think we have like
Good. Okay, you're fancy. Yeah.
Okay, so there was another technique that's very old called ultramicroscopy, where light is shined and then scatter light goes in other directions, and then you can detect it. And
Right. That's
also see.
That's basically dark field microscopy. I'll say I'll connect to that in a second. Okay, so this is kind of the summary of what basically you can do using interferometric detection of scattering. I told you about a transmission arrangement where you do a standard extinction, but I emphasize that that's also an interferometric detection. So you have an object, you focus on it, and then you detect in transmission. You can do that with a point detector. You can do it with a camera. So that would be the wide field version of it, and this would be a confocal version of it. And then you can do it in reflection. This is what I just told you. But you can also have an external reference if you want to do interferometry. So this becomes a little bit more like holography, that you come with a reference and beat it with a light that has interfered with your object. This is the mode that is most common. It's kind of quite similar to this one, except that it's an imaging mode. It's a wide field mode. You illuminate a large area and collect all the light that is scattered back, reflected back from the substrate, and beat it with the light that's scattered from the particle, and then you get an image on this. Now, these methods, they are all kind of sharing the common feature that there is an interference between a reference and light that was scattered by a nanoparticle. And in principle, they're very similar and very closely related to a whole family of interferometric microscopy that in a way date back to the 19th century. So if you read the literature, already in the 19th century, people were doing interferometric microscopy. Except that at the time, they were looking at large objects. So the thing that is really new now is that we're pushing the sensitivity limit to detect smaller and smaller nanoparticles. But the act of doing this type of experiment is common to all of these different types of microscopy. So if you're doing phase contrast, you know you're doing interference. Differential interference contrast, interference reflection microscopy, quantitative phase imaging, digital holography, they're all basically writing the same equation, except that now our object is a nanoparticle, so we can talk about Rayleigh scattering type of thing. And this is where your comment to dark field comes in. Dark field detects at an angle that goes off axis, away from the illumination. So you don't pick the reference intensity or the reference field. You only detect the intensity of the light that was scattered by the object. And I told you that the polarizability proportional to volume, which means that intensity is proportional to volume squared, or the diameter to the 6th power. So now you see that if you were to detect via dark field, your signal would go down very rapidly as you go, for example, from a 100 nanometer particle to a 10 nanometer particle. It would be 1,000,000 times weaker. So the dynamic range that you need to do dark field microscopy is huge, and basically your detectors are not capable of doing that. If you were to detect the extinction term, then your dynamic range can be, in that case, 1,000 times less. But in terms of the shot noise limit, you probably all know that there's only a factor of 2 improvement when you do a homodyne or heterodyne type of detection compared to a normal absorption detection. All right. So that was kind of the basics, and now I'm just going to show you a whole bunch of applications that became possible because we could do this. I showed you that we detected single virus, and a single virus is a pretty small object, especially if you realize that the index of refraction of a virus is only of the order of 1.6. It's in water, 1.3. So the index contrast is actually not that large. So the fact that we can detect light from a 50 nanometer particle in such weak index contrast shows that the method is extremely sensitive. But we want it to be even more sensitive to detect single proteins. And we kind of pushed things a little further and managed to demonstrate that in 2014. And basically, I'm showing you the gist of that work here. If you were to look at a cover glass that is maybe functionalized in your iSCAD microscope in reflection, then you would see something that is kind of speckly. The contrast here is only of the order of 1%. It's not very large, but it's speckly. And it's speckly because you're sending laser light, it's coherent light, and your surface is not perfectly atomically flat. So it has a slight roughness, a slight index of refraction modulation, and that leads to a speckle type of background. That makes detection of very small particles on top of this very difficult because you can't tell whether one of these is a particle or is just a speckle. But the point is that the speckle is actually a pretty constant background. It's a property of the substrate, it doesn't change with time, which means that if you were to record 2 images consecutively of the same object and subtract them, you could mask the whole thing and flatten your field. Now, if between these 2 frames, proteins are added, we can really get to the signal from individual proteins just by a simple subtraction. Now, the process is shot noise limited, which means that if you want to detect very small signals, you have to integrate long enough. So if your protein is large, you can do a single frame subtraction. And if your protein is very small, then you have to batch some of these frames, for example, 100 frames, and then subtract it from the next 100 frames, and to detect smaller proteins. But that's a slight kind of technical detail. The point is that in a very simple manner, you can be sensitive enough to detect scattering from individual proteins. And this is now very interesting because it gives you a single protein kind of sensitivity using light and without using fluorescence. Here are examples of detecting large proteins. I mean, these are still actually small proteins, but the largest that I'm showing you here, 340 kilodaltons, and then 180, 150, and 65 kilodalton. And you see here that the shadow here is quite strong and the shadow here is kind of weak. And that's because the strength of the signal was proportional to the polarizability. And as your particle becomes smaller, the polarizability becomes weaker. What was interesting was that we could show that the signal that we're getting is essentially linearly proportional to the molecular weight. So the molecular weight is telling you how many basically atoms there are, what's the mass of the protein. And it turns out that because of this proportionality, it means that the proportionality to the volume is translating into proportionality to mass, which becomes very helpful because then you can now basically weigh proteins by doing this kind of an optical measurement. So you're not only counting how many proteins are coming, but you're also figuring out how large the protein is. More recently, we showed that we can push the sensitivity down to proteins as small as 10 kilodalton. And what we do here is basically the same experiment, except that we're using machine learning algorithms to pick out small signals on top of noisy backgrounds more efficiently than we could do with our simple background subtraction that I showed you. If you're interested in that, you can consult this paper, but I'm not going into details of that. So you see now that our ladder is even much smoother and better than before. Again, large protein, 220 kilodalton down to something of the order of 10 kilodalton, and they all line up quite nicely as a line. Now, this method is now known even as mass photometry. This was the result of hard work of my former postdoc, Philip Kukura, who is a professor at Oxford University. And he and his team pushed this to show the first time around 2018 that this linearity really works as a kind of a ladder to measure protein mass. And in particular, it's very interesting to show that you can tell the difference between a protein and a protein dimer and a protein trimer and tetramer. And the point that, for example, I didn't know myself when we started this research is that when you buy a protein sample and dilute it, it's never completely always single proteins, but these proteins like to aggregate in different ways. And people who do mass spectrometry and doing protein research actually quite often are bothered by this because they don't know exactly what's happening. And this method now allows us to learn about proteins in their native environment without having to take them into vacuum and doing mass spectrometry.
Depend on the nature of the protein? It's just a matter of the size?
That's basically, to the accuracy that we have right now, seems to be the case. At some point, these things must break down. But at the accuracy of a few percent that we have, this seems to be the case. And also the other question that usually people ask, and it's a good question, is the shape of the protein, because that goes into the polarizability in principle. And all of that doesn't seem to matter that much right now, although we haven't looked at every single protein yet. So this could come out, but you need a little more, let's say, precision in terms of making those measurements. Yes.
Within the object that's being imaged can one utilize the fact that there are different frequencies that are interfering to resolve it?
Absolutely. So this is all done very off-resonant. So we're using blue or green light, whereas the first absorption of the protein is in the UV at 200 something or 300 something. If you use the resonance of the molecule, then you get sensitivity to that, kind of the same way as I showed the single molecule absorption experiment right in the beginning. There we were doing the experiment right at the absorption of that molecule. The difficulty, but this is, I think, completely viable, is that doing these experiments in the UV is more challenging than doing them in the optical domain for the usual things of detectors and objectives and all of that. But you would have that advantage.
Instead, this to like a biochromatic
Yes,
beam and get a similar advantage.
that's right. So we show that as well in that paper in 2011. You can look at the same molecule at two different wavelengths, and that way you can do a little bit of a background kind of suppression because of the spectral properties of your sample. Yes. All right. So again, I mean, this method is now commercialized. There's a company called Refine that sells these instruments, and I've visited many groups who own these devices. So what we did at about the same time was to try to go more towards medical kind of fundamental medical research. And we were trying to look at the very interesting problem of secretion. So when you have a biological cell, this biological cell is constantly breathing and doing its work. And part of what it does is communicate with its environment. And this communication is a chemical exchange. So in particular, a lot of cells secrete proteins and vesicles constantly. And we wanted to look at the secretion of these proteins directly by putting a single cell on a substrate and parking the field of view right next to it. And now as the proteins secrete, in real time, we can watch them basically parachute down on the substrate right next to it. And this movie here gives you a sense of what's happening. So every time that you see a shadow, that's a footprint of a single protein that is landing. And you see that they come at different times and different places. This is just a little field of view at a few microns away from the cell. So by doing this, you get a sense of what the cell is doing over a very long time. And if we wait long enough, you will see here that hell breaks loose if the movie goes long enough. At this point, the cell lysed. That means it kind of exploded because we were treating it in terms of pH a little badly. And but this was something like 30 minutes afterwards. So over a period of 30 minutes, we had precise information about how many proteins were coming out at what time. Now you can take the proteins that are coming out because you can measure the contrast, and then you know that the contrast is related to molecular weight. You basically can get a histogram of the type of things that are coming out. So this was the histogram that we saw. We knew from the literature that this cell was secreting a protein called IgG. And the IgG contribution is this much, but there was more stuff that was being secreted. And we didn't know at the time what all of this was, but we could tell that there's that much stuff at that kind of a mass distribution that was coming out. So this was a very simple demonstration a few years ago. We were pursuing this type of thing, but this was at the single protein level. It turns out, so one of the fun things about doing this type of thing as a physicist, kind of getting into a field that you're not qualified to work on, is that you're constantly learning. And so one of the things that we learned at some point was that there are there's a zoo of objects that are being secreted. So not just single proteins, but proteins that are encapsulated in vesicles. So vesicles or liposomes are objects like this. So you have a lipid kind of bilayer, kind of like a soap bubble. And this could be empty, but usually out of the cell, this is never empty. But it could have proteins, DNA, RNA inside, but it could also have proteins that are embedded in the membrane. And then you could have a so-called lipoprotein, so proteins and lipids all mixed up with each other. You could have all kinds of objects, and of course, virus is one of them as well. So all of these things could be secreted, and they're nanoparticles. And in fact, they're quite large. They're usually in the range of a few tens of nanometers to a few hundreds of nanometers. So we realized that all these years, we could have detected these things extremely sensitively, better than anyone else could do. So at some point, we actually did this, and this paper came out a couple of years ago. And it is showing something that is a spoof on an existing method. And the existing method is called nanoparticle tracking analysis. This uses dark field microscopy. So you take a suspension that has nanoparticles in it, and you illuminate it, for example, like that. And these nanoparticles scatter light, and you collect the scattered light at an angle. And then you can see how individual nanoparticles are jiggling around. Because you're seeing individual nanoparticles, you can extract the trajectories. So when you have the trajectory of them, you can do an analysis on that trajectory and figure out the diffusion constant. The diffusion constant tells you basically something about the Brownian motion of this object. And the diffusion constant is related to the size of the particle according to this formula. So this is Boltzmann constant, this is temperature, this is viscosity, that's pi, and that's d. D is the size of the particle. So by doing this type of measurements, figuring out d from the trajectories, you can figure out the size of the particle. This is the commercial device that you can purchase as well, and is quite helpful. But again, because it does dark field microscopy, its signal is proportional to the square of v, or 6th power of the particle diameter, and its dynamic range is not very good. So usually you're not very sensitive to smaller particles. What we have done is basically done the same experiment, except with iSCAD instead of dark field. So we're doing the same thing, but now we illuminate and then detect in reflection. And this is the kind of an interference pattern that we get. And again, we establish the trajectories and figure out the diffusion constant. But now, in addition to the diffusion constant, we also have the strength of the iSCAD signal, which tells us the polarizability. And that is also related to the size of the particle. So we get two independent measures of the size of the particle, and we plot them in this kind of a scatter plot. So this is derived from diffusion, and this is derived from the iSCAD signal. Now, in this experiment, we had mixed 3 different types of nanoparticles, 15 nanometer, 20 nanometer, and 30 nanometer gold nanoparticles in water. And you can see that we find 3 different populations in this kind of a scatter plot. Now, if you were to put the same sample through NTA, which use dark field, this is the kind of a signal that you would get. So you're not resolving these 3. And some of you might be familiar with dynamic light scattering, which is also a scattering experiment, but it's an ensemble experiment and does a statistical analysis of the light that is scattered. And this is the signal that you would get from that. Again, you're not resolving the 3 populations in this method. So this is right now very exciting, and we're trying to extend this to all kinds of different applications. These are other examples of how we demonstrated kind of the power of this technique, going to the size regime and the combination of sizes that are not accessible to previous methods. To close this part of kind of more biologically inspired work, I show you now how we go away from detecting isolated nanoparticles like proteins or vesicles or viruses to really a crowded region of a cell, of a live cell. For a long time, we were bothered by the fact that if you take a cell and illuminate it with coherent light, you get a lot of speckle. You see almost nothing because it's full of speckles. And of course, in the cell, there's also a lot of dynamics, which means that you get a dynamic speckle that looks like a nightmare. What we realized at some point was that if we do our experiment in the confocal mode, we actually don't have this problem. And here is now a movie that I'm going to run. It's a confocal iSCAT image of a cell. And the structures that you see here are the so-called endoplasmic reticula. These are nanotubes, if you want, of about 100 nanometer diameter. They're soft, kind of tubular things that are dynamic. They're constantly being generated and ripped apart and moving and everything. And because they're so kind of scattering weak, usually in a microscope, you don't see them. And people do fluorescence microscopy to be able to visualize all of these things. And now you can see that a simple kind of scattering microscope can visualize all of this. And so you can see the dynamics of all of these guys. But you also, of course, see other particles that are moving around. These are larger particles or agglomerates, and some of them are smaller vesicles that are moving around. Sometimes these vesicles are carrying proteins. So there's a whole kind of rich landscape that is very exciting to investigate. To kind of bring it back to the pandemic and the virus situation, here is an example where we are looking at, this is now a big program that we have in our lab, trying to understand the virus cell life cycle, because it turns out that maybe you also noticed that during the pandemic, we actually didn't know much about viruses, although viruses have been around for years and years, and virologists have been working on them, and you quickly found a vaccine, but we still don't know how a typical virus comes and interacts with the cell and how it goes in and how it's reproduced and comes out, how it infects the neighboring cell. So a lot of these steps are actually still unknown. And one of the things that we would like to do is we want to, through microscopy, learn more about what's happening. So this cell was infected, and these are viruses that have been generated in the infected cell. We infected them. And what I'm just going to show you briefly is how these viruses are now being moved around with the so-called actin tail. And by visualizing these viruses now without the need of fluorescence in this kind of a label-free microscope, we are able to look at the development of the cell and of infection over long time scales and with the kind of precision that has not been accessible before. Because if you want to do this type of experiment with fluorescence, you have to genetically manipulate your sample, and your fluorescence gets bleached and all kinds of difficulties that you have. But we can do this type of experiment over hours or over days on the same cell and really chase every single virus and see where it begins and what happens to it at the end. So we're very excited about this. So I would like to thank all the people who have been working on this project from the very, very first iSCAT signal that we saw when we were still at the University of Constance, the people who worked in Zurich. Some of them are leading their own groups by now, and the people who are working in the lab today. So this was the part that had to do with this biologically motivated work and microscopy. The physical setup is very simple. I encourage you to test this. You can do this also in a student lab. It's very simple to get the first signal. If you want to get to very high-end type of analysis, the actual image processing becomes a little more complicated, but we're building an open source platform and trying to put as many of the things that we know, or basically everything that we know, we somehow formulate them so that others can use them. And if you have questions, you're always welcome to contact us. Now, I started the talk with a single molecule, but that was a molecule that was at room temperature, and molecules at room temperature are not that they're still quantum objects, but they lose a lot of their quantumness because of the interaction with phonons. But we can go back to that fundamental question of what happens when you illuminate light on a single molecule. I told you that the cross-section is of the order of lambda squared divided by 2. Now, it turns out that we showed in this theoretical paper a long time ago that really, if you were to mode match the incoming beam with the beam that is, or with the light that is scattered by the molecule, then you can get 100% extinction from a single molecule, okay? And again, that kind of makes sense because I told you the cross-section is of the order of lambda squared divided by 2, and you also know that you can focus light down to something of the order of lambda squared divided by 2. So from that sigma over A argument, it makes sense that the effect of a single molecule or a single atom should actually be huge, right? Not that 10 to the minus 6 that I told you. That was because it was at room temperature. But if you go to a vacuum or you go to an atom that is completely unperturbed by its environment, you should be able to get essentially 100%. But for that, you have to match the incoming mode so that your interference becomes perfect. So anyhow, so that's from a theoretical point of view. So it's not a ridiculous thing to do. And we started doing this type of work now almost 20 years ago. And to do that, we have to go to low temperatures. So we're doing this work at about 1.5 Kelvin, because at that temperature, these molecules behave like isolated single atoms. The interaction with the matrix in a kind of a hand-waving way is frozen out. And you get really down to a cross-section that is comparable to lambda squared divided by 2. And this is really the kind of, it's in a simplified way, this is the experiment that we're doing. We're building our own microscope objective in a cryostat because you can't take a standard microscope objective and put it in a cryostat. It kind of breaks. But you can make a simple high NA objective by taking a solid immersion lens, a kind of a half-ball lens, and a simple A sphere, and put them together, and now you have a high NA objective. And we prepared the molecules on this flat surface, and then you have another A sphere to collect the light. And if you now tune the frequency of the laser through the transition of the molecule, you see that when the molecule is resonant, where the laser is resonant with the molecule, you get something of the order of 10% extinction. So it's not 100% yet, but it's much better than 10 to the minus 6 that I told you before. And I'll tell you in a second why it's not 100%. But this was very exciting. It was the first time that, someone showed that extinction from a single quantum emitter can be really that large. And we've kind of exploited that effect in a number of studies that I won't talk about because they're now kind of older. So this is the gist of it. Now, if you think about the molecule is not a two-level system, but it has vibrational levels for the ground state and for the excited state. And that's where fluorescence comes from, right? So you excite the molecule from this level to this level. That's what we usually do. But when it decays, it can decay via the same channel or via these other channels, which have a lower lower frequency or larger wavelengths. So they're red-shifted. This is the fluorescence channel that one usually detects, but we showed that you can also detect it by doing basic extinction. So this is the extinction signal that I showed you, and simultaneously we can record these photons, and then you get the fluorescence signal. So a little bit about the system. This is a very special system, molecular system. This is a family of so-called polycyclic aromatic hydrocarbons. These are basically kind of, if you want, a little bit like you take a sheet of graphene and you go very carefully go around and cut with atomic precision. This is what you get. These are all the corners are carbon and then extended with a hydrogen, passivated with hydrogen all around. But depending on the shape of the molecule, the transition is shifted a little bit somewhere in the visible or near infrared. So these are the actual molecules that we use. Now we embed them in a molecular crystal that is usually made of these molecules. And the molecular crystal has a band gap that is more in the UV. So it doesn't absorb, so it's transparent for visible light. But these molecules have their transition somewhere in the visible. So we're talking to these guys, and this is just a matrix to hold the molecule. Now, the interesting thing is that when you go to temperatures below 2 Kelvin, the transitions of these molecules become extremely narrow. They become as narrow as a few tens of megahertz. You are probably not familiar with these units as much, but it's the same kind of bandwidth that you have if you had an alkali atom suspended in vacuum. So although the molecule is embedded in a molecular crystal, in a matrix, in a solid, its interaction with the environment is essentially frozen out to be no longer important. So you really see the intrinsic transition from ground state to the excited state. That's basically what this line width is telling you. And this is a so-called Fourier limited line, which means that the line width is really just the inverse of the lifetime that you would get. That means there is no extra dephasing that comes in to broaden the line. All right. So work on molecules has been very slow. We have been probably the kind of most persistent group over the past 20, 25 years to try to do quantum optics with molecules. But there is now a kind of a revival, and people are finding it interesting, and lots of people are working in from different directions. So if you're interested or curious about that, we have a review article that was published a couple of years ago, so I can recommend reading that. So generally, the situation is as follows. You have these molecules that are embedded in a crystal, but they're not all completely identical because the crystal is never a perfect crystal. So you can think of the crystal having slight defects or slight variations, and that means that the frequencies of individual molecules are slightly shifted from each other. And that's what gives us the ability to detect single molecules, because although, for example, your laser beam would be talking to many of these molecules or shining on many of these molecules, at any given frequency of your laser, only one of them is resonant. And as you tune the frequency a little bit, you see strong lines popping up. You have a question?
And can you see cooperative effects?
Right. I'll come to cooperative effects at the very end of the talk if I get there. I'm been very slow, so I probably have to wrap up in a few minutes. Yeah, I'll try to do it 5. So if you zoom into one of these things, then you see that there is this narrow line that I told you about. But you also see that from a kind of a data storage point of view, you have an incredible kind of capacity there, because you have all these molecules that you can have within a very small frequency range. These are only a few hundreds of gigahertz apart. All right, so I told you about this situation before, and the fact that we go up on this transition, and then, but when you're up there, you can come down like this, but you can also come down like that. And this gives us the fluorescence channel, but as far as the coherent interaction with light is concerned, these channels are not good, because these levels have a very short lifetime, and these levels are strongly coupled to the matrix. This level is not, but these levels are strongly coupled to the matrix. And that means that this channel spoils the coherence of my interaction. And the so-called branching ratio is about 30 to 50%. So it's not that bad, but it's not 100%. That means that if I'm up here, about 30% of the time I come down like this, but about 70% of the time I come down like that. And that 70% kind of spoils the game. That's why I don't have that 100%, there's a chance to have 100% interaction or scattering efficiency. Now, the way to go about this to fix the problem is to enhance this transition so this transition becomes faster. It happens before this gets a chance to do its job. And the way you enhance a transition is to put your emitter in a cavity. This is something that has been a field of research, so-called cavity quantum electrodynamics, for the past 40 years or so. The essence of it is that if you put a photon here, you bounce the photon back and forth, and every time that it goes around, it gets a chance to interact with the molecule. Now, if it goes around by basically finesse times, you get an enhancement by the cavity finesse. So the figure of merit that you have for the enhancement is the finesse, but then you also usually lose because the mode area in a typical cavity is larger than you would have if you were to focus in a diffraction limited microscope. So, but as long as this factor is larger than 1, you're winning in terms of enhancement. And this factor you can rewrite, and you can actually show that it's the same thing as Q over V, which is a more common way of understanding cavity enhancement or Purcell effect and such. It's also related to cooperativity, but basically what you want to do is you want to have a cavity that, and if you increase Q or lower volume, you are doing well. So this is kind of the name of the game that we wanted to play about 15 years ago when we started this project. We wanted to have a cavity that has a very small radius of curvature on one side in its mirror so that we can make it very small, to make that mode volume very small. And then you would have a flat mirror on one side so that you can put a small crystal with these molecules in it. And this would be our cavity. And then you just use a lens to couple into it and all of that. So the way we go about this is we can take an optical fiber or the end of an AFM tip, and we use focused ion beam milling to make micromirrors. At the end of it, this is a kind of a profile that you would get, and then you can coat this in a multi-layer way. This is what we do at a company, and this gives you a very high reflectivity. And now by bringing this tiny mirror with a radius of curvature of only a few microns to within only a micron or 2 of the substrate, we make a microcavity that has a very large finesse because it has a low volume. And this is some of the early experiments that we did. This was published in 2019. This is the resonance of a cavity like that in transmission and reflection. Now, if you tune the resonance of the cavity to one of the molecules that is in that crystal, then you see that the resonance changes completely. So you're burning kind of a hole right in the middle of the transmission because there's one single molecule in the cavity. In a kind of a simplest way of thinking about this is that you've changed the resonance condition of the cavity because you put one molecule in it. And usually you change the resonance by putting something of a certain index. You shift the resonance. So the index of a single molecule, if you want, is large enough to kind of change the resonance condition. Or you can think of it as an extinction of a single molecule now that is being enhanced by this cavity. And now you're getting this almost 100% extinction from a single molecule. What we could also do was to show that we could replace the laser by a single photon stream, so that instead of sending lasers onto a single molecule in the cavity, you send individual photons onto it. I'll skip this part because I want to get to the more interesting end without keeping you too long. And you might know that in this cavity QED type of experiment, you have a so-called weak coupling regime where you have this Purcell enhancement that I told you about. But you can also go to a strong coupling regime. That's where the situation like two coupled pendula, and now the two resonators. One of them is the molecule, the other one is the cavity. Each one of them has a resonance. That means that it can store the photon in it. But now if the two kind of storage situations, capacities become comparable, the photon can bounce in the cavity, come back to the molecule, and go to the cavity and come back to the molecule. So in the weak coupling regime, the photon comes, interacts with the molecule, and leaves again. But in the strong coupling regime, the photon can bounce back and forth many times and give this energy to the cavity and come back to the molecule. And as it does that, in the time domain, you begin to see oscillations. And we showed a few years after the first work, but by now already 3 years ago, that we can also enter this strong coupling regime with a single molecule. Now, if you do that, you're in a regime where you have a very strong nonlinearity. So nonlinear optics is, of course, very interesting for all kinds of reasons, but you also know that optical nonlinearity is a pretty weak effect. It's a higher order effect. So usually you need a large crystal, you need a lot of intensity to be able to see much. But in fact, a single atom or a single molecule is a pretty strong nonlinear system. But if you just put a single molecule in your laser beam, usually it doesn't do much. But in this case, a single molecule is going to do a complete switch. And to demonstrate that, what we're showing here is that you have a molecule in the cavity, as I just showed you. And now I'm having two beams, two laser beams that are detuned from each other only by about 300 megahertz. So one of them is completely resonant with the molecule in the middle. The other one is a little off. And if I were to look at the resonance, if the so-called, the one that is off, that I call pump, is off, then I see this. But if I turn the pump on, then I change the transmission of the system. So the transmission goes from 0 all the way to 1. So by adding a second laser beam, I'm changing the transmission of the first laser beam. And that's basically an optical switch that I have. And the interesting thing is that this is happening when there's only, on average, one photon in the pump. So the pump is called pump, but it's really not strong at all. It's just a single photon kind of beam that I have. So this is all very interesting because it really gets you to a regime where you can play with single photons and single molecules in a very, very efficient way. There are lots of kind of detailed, subtle issues that prevent us from immediately turning this into some kind of a device. But to kind of bring us this far, this has been very exciting to have that much control over the interaction of single molecules and single photons. Now, the last thing that I want to tell you, but I'm going to cut it very short, is really just a question of cooperativity. Basically, doing work on single molecules and single photons was very interesting. It occupied lots of groups and lots of systems for the past 20 years. People have worked on single quantum dots, single atoms, single color centers, single molecules, different ways of generating single photons. But I would say that although there's still a lot to be done, the field is pretty mature. You're not going to impress too many people if you have another single photon source. But scaling up is kind of the bottleneck or the next challenge, is how to have many single photon sources or many single quantum emitters and to make them interact with each other very efficiently, to build something like a quantum network that actually works efficiently. This is a very interesting field of research, but also very difficult to do. Part of it has to do with the fact that already one photon and one quantum emitter, it's difficult to get them to interact very efficiently. And if you want to build a network, you lose exponentially if you have losses at every node. Nevertheless, you try hard, you try to kind of develop the technology, and the kind of things that I showed you would be the building blocks. And now you have to kind of put the building blocks together. one way to do this is to put it on a chip so that you can scale up. So we're now building kind of photonic circuits. This would be, for example, a microdisc resonator with a certain resonance, and then you have different waveguides to couple light in and out. And then you can put molecules on top. Preparing the samples and all of that is always a challenge because you have different materials, and material science always, or so far, has always killed everyone. You know, if you talk about quantum dots or color centers, they're all killed at the end by material science issues. It's also a big problem for us because we're putting organics on inorganic kind of circuits, and there are lots of issues that you have to solve. It's not a dead end, but it's a very slow process because physicists don't know material science. And so you put molecules and try to couple evanescently. And for example, you know, again, you see that there is a single molecule coupled to the resonance of this microresonator. And now you can put microelectrodes and tune the frequencies of different molecules so that you can bring them to the same frequency so that they can talk to each other via the mode of the optical resonator. And this is the kind of thing that you would do. In this case, you see that you have lots of molecules coupled to the microresonator. You take two of them, and then by tuning them, you can bring them somewhere around here to have the same frequency. And we showed in this work, yeah, it's already appeared. This is an old slide. Appeared a couple of months ago in Optica. And we go through the details of how two individual molecules are identified and then brought together in frequency, and you can show that the light that is scattered from each one of them actually interferes constructively in the forward direction and destructive in the backward direction, that kind of thing. And the particularly exciting work that I'm not going to tell you anything about today is something that we're trying to write up, but it's done in this geometry that I told you about before. So you're in an open microcavity, and there we're also getting to a situation where light that is scattered from one molecule is going to be interacting with another molecule that is in the common kind of in the same mode. And I just want to flash two fairly recent papers that I'm not going to have time to talk about. If you're interested in more near field type of experiments, this we think is an exciting tipless kind of arrangement to bring a quantum emitter, for example, in the near field of an optical antenna. And this is work that was published just a couple of weeks ago, where we show that in a kind of a fluidic arrangement, you can use concepts of optical antennas to collect more photons and therefore do more efficient single photon, single molecule type of experiment. And with that, I would like to also thank the people who are working in the quantum optics part of the group right now. Lots of people have contributed in the past. They wouldn't have fit in one slide, so I don't show them. But these are the people who are currently working in the group, and we would love to hear from you if you're interested in doing postdoc work in any of these different directions at the Institute. And with that, I would like to thank you for listening. I'd be happy to answer any questions.
Yeah, thank you very much for this very fascinating talk. I think we still have time for one or two questions, and the speaker will also be around later. Before we take the questions, I should also mention that there is an information session tomorrow about studying in Germany, especially at the Max Planck Institute for the Science of Light. And this is tomorrow at 12, I think, in the large lecture hall, and pizza will be provided. So, okay, so I think we have time for one or two questions. No questions? All questions answered? Then I have a quick question. You have a question now.
I have a question about the molecule experiment that you were showing. By the way, this is super cool. So, were the molecules actually sitting on top of the resonators in that case? or were
In
they
the first case, they're sitting on top of the resonator, on top and next to it. So you have a disk, it's a micro disk of titanium dioxide on a glass substrate, and then on that we grow an organic crystal, and that organic crystal includes the molecules, so they're coming close to the boundaries of the resonator.
So I'll ask my follow-up questions. Thank you.
What are the spin properties of these molecules? Do they have any?
Yeah, so these molecules don't have any spin, which is, again, a material problem. So I mean, there are molecules that have spin. Their spectral properties are not as nice as these ones. And so far, we haven't worried about spin. There are a few people who are trying to push that. The molecules have a triplet state, which would have a spin, but it's not in the ground state. So color centers have a nice situation that the ground state is a triplet state. But here we are looking at a few different situations where triplet gives us spin properties, but those are normal triplet states that are at a higher frequency.
Okay, so then let's thank our speaker again. So we have this certificate for you, and we thank you again for this very nice talk.
Thank you very much.
Thank you very much.
Thank you.